Alright, so this video is going to be me going through dihybrid crosses one more time, and I'll have an example that I'll work through at the end. So again, we've been solving monohybrid crosses so far where we're only looking at one trait at a time. So what's the chance that the offspring in a cross are going to have round seeds or wrinkled seeds? Now we're moving into dihybrid crosses. And dihybrid crosses are going to look at the chance of inheriting two traits together. So what's the chance of the seed being round and wrinkled? So we're looking at the trait of seed shape as well as seed color at the same time. So when we are looking at dihybrid crosses, remember di means two, and we're looking at the chance of inheriting any two traits together at the same time. We're going back to Gregor Mendel. Gregor Mendel did monohybrid crosses, and he also did dihybrid crosses. So when he looked at his pea plants, he could look to see if the seeds in the pea plants were round or wrinkled, so the shape of the seeds, but he could also look and see if the seeds were yellow or green. So in this case, our key is just expanded a little bit, where capital R equals round, lowercase r equals wrinkled, round versus wrinkled, capital Y is going to equal yellow for seed color, and lowercase y will equal green for seed color. So we're looking at both traits at the same time. So remember, every organism has two letters for every trait because they are diploid. Two copies of every trait. So they have two letters for seed shape and two letters for seed color. So in this case, they're going to look like two letter R's and two letter Y's. When Mendel did his experiment, remember, he always started with true breeding plants, which are homozygous plants. Homozygous for round and yellow is going to be capital R, homozygous for round, and then homozygous for yellow, which is capital Y, capital Y. The opposite homozygous would be homozygous for wrinkled, which is lowercase r, and then homozygous for green, which is lowercase y, lowercase y. So he crossed these two pea plants, and he noticed that all the offspring were then heterozygous, because this parent gave a big R and a big Y. This parent gave a little r and a little y, so the offspring was fully heterozygous, having a big R and a big Y, and a little r and a little y, fully heterozygous. He crossed two yellow round plants, looked at the offspring, and he had all types of combinations. He had some that were round and yellow, some that were round and green, some that were wrinkled and yellow, and some that were wrinkled and green. This showed up as a 9-to-3 to 3-to-1 ratio. So in this case, we have four different possibilities of what the offspring could look like, just because there's more combinations when we're looking at two traits. This led Mendel to his second law of heredity, which is the law of independent assortment. The law of independent assortment states that you get and inherit each trait independently of the others, meaning that just because a seed is round doesn't mean it's going to be yellow. It can be round and wrinkled. Your traits aren't inherited together. Just because I get a gene for brown hair doesn't mean I'm also getting a gene for brown eyes. I can get any combination for any traits because each trait is inherited independently from other traits. Now, one thing about dihybrid crosses is that we're going from our small monohybrid 4-square Punnett square to a 16-square Punnett square. Now, dihybrids are very, very tricky. So you're never going to be asked to set one up. You might be asked to identify the genotypes of the parents. You might be asked to identify the genotype of the offspring once the square has already been set up and given to you. So in this case, any square you get will already be set up. You just need to know how to fill in the boxes. You'll also need to know how to figure out the phenotype of the offspring. So if I was to give you the letters here, you would need to tell me what it's going to look like, given a key. So it's very similar to what we've already done in the past, except we're looking at two traits at one time. So in this case, and don't worry, we're going to do more examples here in a second. In this case, when we're filling in this missing box here, what we are doing is we are pulling the letters. So for this missing box, I'll circle in blue. We are pulling this letter from parent 2 and this from parent 1 to fill in this box here. So the two R's that this individual receives is a big R and a little r. Big R, little r. They receive a little y and a big Y. Big Y, little y. And then we just lose our key. For the R's, big R, little r, having a big R means it's round. Big Y, little y, having a big Y means it's yellow. So this one is round and yellow. For this box here, let me use a different color. I will circle in orange. For this box here, we're going to have this from parent 2, these alleles from parent 1 going together. So for the R's, it's going to have two little r's. Two little r's. It's going to get a big Y and a small y. Big Y, small y. Little r codes for wrinkled. Big Y, small y codes for yellow. This one is wrinkled and yellow. And then for our last box, my last color, I will circle in red. For this box here, these alleles from this parent and the ones up here from this parent will combine. So for letter R, it's going to have a small r and a big R. Big R, small r. And a little y and a little y. Little y, little y. Big R, small r codes for round. Little y codes for green. And that's what you need to do. So at this point, I'm going to keep going. I'm going to have more explanations for you if you need it. If you totally get it, you can stop. But I'm going to keep going. I'm going to do this practice at the bottom and one more example just in case you need it. Again, if not, feel free to go ahead and stop watching the video. Totally up to you. So this practice problem here, we have a dihybrid cross. In this case, we have two traits to consider. Tall versus short, so we're looking at height, as well as color, blue versus white. This is looking at flowers. Our Punnett square for our dihybrid is already set up for us. All we have to do is analyze it. So let's go through our questions. Question number 1, it says, what is the genotype and phenotype for box 1? Our genotype are our letters. So genotype are our letters. For box 1, we're going to pull from parent 2 here. This one comes here. And from parent 1 here, and it'll come together. For T's, it'll have a capital T and a lowercase t. For B's, it's going to have a lowercase b and a capital B. Remember, capitals are always written first. So the genotype for this one is big T, big B, little b. Then it also asks for the phenotype. The phenotype is what it's going to look like. We just do our letters separately. We do our T's first. For our T's, big T, small t. Big T equals tall. So this is a tall plant. Now we're going to look at our B's, big B, small b. Big B equals blue. So this is a tall blue flower. Great. Now for number 2. Number 2, what are the genotype and phenotype for box 2? I'll switch colors. Let's go to blue. So for box 2, when we're looking at our genotype, this parent is going to give here. This parent is going to give these letters, and they will combine. For T's, there will be a big T and a small t, and two large B's. So our genotype is going to be big T, small t, big B, big B. Then our phenotype is what it's going to look like. Big T, small t, big T equals tall, so this one is going to be tall. For big B equals blue, so this is going to be a tall blue plant. All right, now moving to number 3. Number 3 says, what are the genotype and phenotype for box 3? For box 3, we're going to have this parent giving those alleles, this parent giving these alleles, and they combine. For T's, there's going to be 2 small t's and also 2 small b's for box 3. So our genotype is small t, small b. And for our phenotype, that's what it's going to look like. Small t is short. Small b is white. This is a short white flower. And then question number 4, I'll change colors again just to one that's a little darker. How many of the 16 possible offspring are heterozygous for both traits? Heterozygous for both traits it's asking how many are big T, small t, big B, little b. That's 100% heterozygous. Heterozygous for T and heterozygous for B. Conveniently, it also told us there's 16 total. So all I have to do is count up how many are big T, small t, big B, little b. So I'm going to search for them, and I'm going to box them in red. And then we're going to count them up. So those are our heterozygous. I circled them. There are 4. 4 out of 16 are heterozygous. All right, so now I'm going to go through further explanation if you need it. If you don't, feel free to stop watching. This is a second explanation going through the whole process. So in mice, black fur is dominant to brown fur, and straight hair is dominant to curly hair. Cross 2 heterozygous mice. Again, we're never going to ask you to set this up. It's always going to be set up for you. But I'm going to show you where these come from so you'll have a better idea. And I'm also going to show you our key. So I went ahead and created our key. Big B is black, little b is brown, big H is straight, little h is curly. You get that right from the problem. The next step is to find your parents. It said both parents are fully heterozygous. So both of these parents are fully heterozygous. The square is already set up for you. I'm never going to ask you to come up with these. You'll never have to do that. But I want to show you where they come from. So in our organism, parents are diploid, meaning they have 2 copies of every letter. But when they create their gametes to give their offspring, the gametes are haploid. They only give 1 copy of each letter. So along the top, you see 1 B and 1 H, because each parent's going to give 1 B and 1 H. But the thing is they can give any combination of their B's and H's because they're inherited independently. Parent 1 can give a big B or a small b, and it can give a big H or a small h. But there's many combinations of B's and H's this parent could give. It could give a big H and a big B and a big H, represented with here. It could give a big B and a small h, as represented here. It could give a small b and a big H, represented here. Or a small b and a small h, as represented here. That's where these come from. They're all the different combinations that parent 1 could give. Along the side are all the combinations that parent 2 could give. These will differ based on our parents' genotypes. But again, you're never going to have to come up with those. They will always be given to you. But you will need to know how to fill out the square. So given what's on the side, you'll need to know how to fill out what's in the center and also figure out what they look like. So let's practice. For this box here, let me go ahead and number these 1 through 16. That way I can just give you a number and you'll know exactly what one we're talking about. All right, for box number 1, these gametes are going to combine with these. There's 2 big B's and 2 big H's. Using our key, big B stands for black. Big H stands for straight. This mouse will have black straight hair. For number 2, this gamete will combine with this gamete to give us 2 big B's, a big H and a small h. That will lead to big B, which is black. Big H, big H is straight. A mouse with black straight fur. For number 3, we have this gamete combining with this one. There's going to be a big B, a small b, and 2 big H's. Big B stands for black. Big H stands for straight. This one will also have black straight hair. Can't talk and write at the same time. For box number 4, this gamete will combine with this gamete. There will be a big B, a small b, a big H, and a small h. This one will also have black and straight hair. For box number 5, this gamete combines with this gamete to give me 2 big B's, a big H and a small h. That's going to be black straight again. For number 6, we have this gamete combining with this gamete to give us 2 big B's and 2 small h's. Big B stands for black, but in this case, 2 small h's stands for curly. This offspring will have black curly hair. For number 7, we have this gamete combining with this gamete at the top. We'll have big B, large H, small h. Again, this one is black and straight. For number 8, we will have this gamete combining with this gamete to give us big B, small h, small h. Big B is black, 2 small h's is curly. That one will have black curly hair. For number 9, this gamete will combine with this gamete. We will have a big B, a small b, and 2 large h's. That will be black, straight hair. For number 10, this gamete and this gamete combine. Big B, big H, small h. That will be straight black hair. Sorry, I'm writing that one backwards, my bad. Number 11, we're going to have this gamete combining with this gamete. We're going to have 2 small b's, but 2 large h's. Small b is brown. Big H is straight. That mouse will have brown straight hair. For number 12, we have this gamete combining with this gamete. We'll have 2 small b's, a big H, and a small h. This will be brown. Big H stands for straight. This is another mouse with brown straight hair. For number 13, we're going to have this gamete combining with this gamete. We'll have big B, big H, small h. That will be black, straight hair. For number 14, this gamete combines with this gamete to give us big B, little h. Big B stands for black. 2 small h's stands for curly. This one will have black curly hair. For number 15, we're going to have this gamete combining with this gamete. We'll have 2 small b's, a big H and a small h. This is going to give us brown. Big H gives us straight. That one will have brown straight hair. For number 16, this gamete will give us 2 small b's, 2 small h's. That will give us brown and curly hair. Next, it asks for the genotypes and phenotypes of the offspring. So we're going to write, let's do genotypes first. For the genotypes, I recommend just going down the list and writing all the different combinations we see. Genotypes are letters. So my first one has big B, Big H, big H. I also see big B, big B. Big H, small h. There's another one. We could also have big B, small b, big H, big H. Here. We have big B, small b, big H, small h. So I'm just writing down all the different ones I see. We already have that one. Ooh, we don't have this one. Big B, small h, small h. We already have that one. We don't have that one. Big B, small b, little h, little h. We got that one. Got that one. We don't have this one. Small b, big H, small h. We got that one. Got that one. Got that one. And then our last one. So those are all the different genotypes we could have. Let's count them up. How many big B, big H do we have? 1. Just 1. So there's 1 of those. How many big B, big H, small h do we have? 1, 2. 2 of those. All right, big B, small b, big H, big H. 1, 2, 2. How many of these do we have? Fully heterozygous. 1, 2, 3, 4. How many of these do we have? Big, small, small. 1. I'm only seeing 1. Okay. How many of these? Big, small, 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 small. 1. 2. All right. Small, small. Big, small. One, 2. And then there's only one of the last one. And just to check, they should all add up to 16. 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Oh, I'm missing one. Oh, there's 2 of those. Counted wrong. All right, there we go. All right, now we have phenotypes. Phenotype ratios, all the different ways they look. Well, we have one that's black and straight. We have a black and a curly. We have brown and straight. And we have a brown and curly. From there, all you have to do is add up how many of each you have. If you add them up, 9 are black and straight, 3 are black and curly, 3 are brown and straight, and 1 is brown and curly. So that is how you go through the whole process. I hope this helped. Let me know if you still have questions.